While we've already covered a lot of the other organelles in the cell in previous videos, we need to look at the nucleus now. The nucleus is unique because it contains all the cell's DNA, contains the genetic code of your body. So the image you see here is showing the nucleus next to what's labeled as a cisternae. Remember, cisternae is just flat membranes stacked together. This particular cisternae, all of this out here, is going to be the rough endoplasmic reticulum. You can see the little dots are the ribosomes. Ribosomes are going to help out with the protein synthesis. We'll get there in a second. First, looking inside the nucleus, you can see that there's a nucleus and there's a nucleolus. This inside structure is going to be the nucleolus. The nucleolus is a very densely packed area of chromosomes. The nucleus itself is still having densely packed chromatin, but you can see how they're kind of more spread out. There are these wavy lines, which means we're not quite as densely packed, and it allows for the actual transcription of that DNA. Now, hopefully remember, DNA, double strands, we talked about that before. But this inside the nucleus, once the DNA gets unpacked a little bit, it allows for a process called transcription. It's where the DNA can be copied into RNA. And then that RNA can leave the nucleus. It leaves via these actual nuclear pores, those little openings that allow the RNA to leave, but prevent the DNA from leaving. RNA being only single-stranded, it's a little bit smaller, can fit out through those nuclear pores. So the nucleus itself is really a self-contained area to keep the DNA safe inside your cell. But that DNA, that actual genetic code, is what controls everything that goes on in the cell. From making proteins, to signaling messengers, to structural proteins, to you name it. The DNA is really what controls that. So let's look into DNA replication to start with. Now, in the case of DNA replication, first of all, we have to start with the actual chromosome. That's densely packed chromatin. But as you start to unspiral it, you'll notice the chromatin is wrapped around these histones. That's like a sphere the DNA wraps around. The DNA itself is so tiny and so fragile, this gives more structure to it. Once you finally unwrapped the DNA helix, you can see down at the bottom that you have, remember, the two sides. You have the nucleotides on each side, adenine, thymine, cytosine, guanine. It's that ACTG coding that is the genetic coding. That is your DNA. To replicate it, the DNA can actually copy itself, making a new double-stranded helix. But it's not just always simply a copy process to replicate it. Sometimes you're trying to transcribe it into RNA. That transcription is how you send things out the nucleus. So as we go through this transcription and the actual duplication or replication part, keep in mind the structure. The DNA backbone, the actual ladder outside pieces, is going to be that sugar and the phosphate. Sorry, wrong spot. Sugar and phosphate. That makes up the outside rungs, while the nitrogenous base, which is right here, is what makes up the rings of the ladder. So the actual middle parts, this red and blue and green and yellow, adenine, thymine, red and yellow, blue and green, cytosine, guanine, makes up the rungs of the ladder. So as you're trying to create new variations of this, you have to recreate each nucleotide. So make sure the nucleotides match together. So as you start the process, you have your DNA, it's unwound. You have to actually unzip the two sides, separate the two sides. That's where this DNA polymerase comes in. The DNA polymerase, doesn't matter which side of the molecule it's on, it helps to unzip it and then read the actual structure. So the helicase starts it. So that starts the whole thing. That kind of like a wedge that unzips. But then you have these DNA polymerases coming right behind it. You can see how they're DNA polymerases are going in two different directions. That's because you have to make sure you read it in the proper orientation. So you have to make sure you're reading in the direction, otherwise if you do it backwards, it doesn't code the same thing. So when it's reading in the proper orientation here. As it reads, you'll notice that you're putting individual nucleotides in place. All these individual nucleotides are being added in according to its base pairs. So adenine goes with thymine, cytosine goes with guanine. That's for DNA replication. That's how we're making this whole new strand up here. But now, if we want to change up and go into, instead of DNA replication, we want to make a messenger RNA. That's transcription. It looks very similar. We still have the free nucleotides. But now instead of using thymine, we switch that with uracil. And uracil becomes part of the RNA. As that goes through, what you're doing is you're reading along the DNA. And you're making a messenger RNA transcript. If you notice, for example, this top strand, that was the template strand. That's what was read. You'll see then that the A down here matches the T above. The U matches the A. The G matches the C. You're matching and having complementary base pairs. So keep in mind though, that as we go through that, uracil is what matches up to adenine now. Uracil is part of the messenger RNA. But then we're not done yet. That messenger RNA needs to leave the nucleus. It's going to leave through those nuclear pores and head out to either a ribosome of the rough ER or a ribosome that's floating around, a free ribosome. It's those ribosomes that can take this messenger RNA strand, and then they take the messenger RNA strand and translate it. So you can see here this is the messenger RNA, this strip. That messenger RNA needs to start with this AUG. AUG is the start codon. Codon being a triplet, 3 of the actual messenger RNA nucleotides. So AUG is going to be the start codon for methionine. You see it right here. Once that one's read and methionine is attached, the ribosome then reads the next codon, which in this case, the next codon is UUU. As it reads that triple U, it attaches the next actual amino acid. As that second amino acid is attached, the first amino acid forms a peptide bond. What will then happen is the third codon is read. A third amino acid is brought in, and what will happen is that third one will bind a new peptide bond. And you keep adding by reading every codon, triplet, codon, amino acid, codon, amino acid, codon, amino acid. And it allows the entire polypeptide chain to grow. Which means that from our transcription, the DNA to RNA, we then translate from RNA to polypeptide chains. The idea is you're transcribing DNA to RNA because it's still nucleotide to nucleotide, just transcribing it. But you're translating it into a new language when you go from the actual individual nucleotides into amino acids. Because you take a triplet codon, makes one amino acid. So the idea of protein synthesis here. All from that DNA, transcription, translation. So all of this protein synthesis is based off only 20 of these amino acids. So what happens here is, you can see there's 3 different sides of this chart. On the left is first base, top is second base pair, and right is third base. The idea is you take the different letters of what the codon is. Like say AUG is methionine. AUG, which is right here, you can see is methionine. But any of the other ones, and you start looking through them, and you're saying, okay, well, CAC histamine. Or you go through, down here, and GGC is glycine. The idea is those are each individual amino acids and the different types of codons that can control them. So any of those 4 codons, for example, could make glycine down there. 20 amino acids, all based on what is coded from the initial DNA, all based on how the order and chain works. All of these amino acids eventually make a long polypeptide chain. That polypeptide chain can be folded into functional proteins. So a little about how your DNA from your nucleus then can make your proteins and how that transcription translation process works.